After studying this module, you shall be able to know multi-metal zinc enzymes, alkaline phosphatase and liver alcohol dehydrogenase and the reactions catalyzed by them. Understand the role of other metals in these zinc enzymes. Identify the catalytic site of these enzymes and learn about the catalytic mechanism for the reactions catalyzed by enzymes alkaline phosphatase and liver alcohol dehydrogenase.
In the previous module, we have studied about two zinc enzymes, carboxypeptidase A and carbonic anhydrase 2. There are two zinc enzymes, carboxypeptidase A and carbonic anhydrase 2 had only one element, that is zinc in them. In this module, we shall study two more zinc enzymes, alkaline phosphatase and liver alcohol dehydrogenase, which are multi-metal zinc enzymes. We shall study about their structure, active sites and reactions catalyzed by them. We shall understand the role of other metals in these zinc enzymes. There are many zinc metalloenzymes which have one or more metals besides zinc. These metals have a co-catalytic role or a structural role. In a co-catalytic zinc site, the metals are in close proximity to one another and operate as a functional unit. One metal plays the catalytic role and the other metal atoms enhance the catalytic activity of the site. Alkaline phosphatase is an example of an enzyme that contains co-catalytic zinc site. The enzyme alcohol dehydrogenase has two zinc atoms, one is catalytic and other a structural zinc atom. Let's discuss these two enzymes. The first one is alkaline phosphatase, abbreviated as ALP. The homodimeric metalloenzyme known as alkaline phosphatase, that is phosphate monoester phosphohydrolase alkaline optimum, is a hydrolase enzyme with 449 amino acid residues. It removes phosphate groups from many types of organic phosphates such as nucleotides, proteins to provide phosphate required for bone growth. Alkaline phosphates is found in all kingdoms of life, plants, animals as well as in bacteria, with the exception of some plants. It is the first zinc enzyme to be discovered which have a catalytic zinc center that contains more than one kind of metal atom. It has a mass of 94 kilodalton. Each monomer of alkaline phosphates binds metal ions. The metal ions they carry can differ. Although zinc and magnesium are the most common alkaline phosphate, catalyze hydrolysis of a variety of phosphate monoesters. It also catalyzes transphosphorylation reactions in the presence of high concentration phosphates acceptors. Enzymes, alkaline phosphates transfers phosphates to alcohol to form another phosphate monoester. Alkaline phosphates is a membrane bound and found in liver, bone, intestine, kidney and placenta. The liver, bone and kidney enzymes are different forms of same gene product. The intestine and placental forms are isoenzymes. Physiologically increased levels of alkaline phosphates are seen during periods of active bone growth, particularly in infants and at puberty. The optimum pH for the activity of alkaline phosphates is in the mild alkali region.
The overall dimension of alkaline phosphatase is approximately 100 angstrom by 50 angstrom by 50 angstrom, with the active sites being 30 angstroms apart from each other on opposite sides of the molecule. Each active site of the dimeric enzyme contains 3 metal binding sites. 2 are occupied by zinc 2 ions and 1 by magnesium 2 ion as shown in the figure. The 2 zinc ions are 4 angstrom apart and the magnesium ion is approximately 7 angstrom from zinc 1 and 5 angstrom from zinc 2 in each of the subunits. The 2 subunits in the dimer are held together by hydrogen bonds, hydrophobic interactions and a salt bridge. Comparison of the amino acid sequences from a number of alkaline phosphatases shows that the enzymes are very well conserved, especially near the active site. The residues serine 102 and arginine 166 are conserved in all the cases. These amino acid residues interact directly with the phosphate ester. The X-ray crystal structure of alkaline phosphatase complexed with inorganic phosphate was determined by Chem et al. The first zinc atom, zinc 1, has a coordination number of 5 with trigonal pyramidal geometry. Zinc 1 binding site consists of histidine 331, histidine 412 and aspartate 327 residue. Both carboxyl oxygens of aspartate 327 are coordinated to zinc 1 along with imidazole nitrogen atoms of histidines. Zinc 1 is also coordinated to one of the oxygens of the phosphoester. The second zinc 2 has a distorted tetrahedral geometry. Zinc 2 binding site consists of aspartate 369, aspartate 51 and histidine 370 and one oxygen of the phosphoester. One of the carboxyl oxygens of aspartate 369 and aspartate 51 coordinate zinc 2. The nitrogen atom of histidine 370 is also coordinated to zinc 2. The magnesium 2 ion has a slightly distorted octahedral coordination environment. Second carboxyl oxygen of aspartate 51 is coordinated to magnesium 2. Magnesium 2 is also coordinated to 2 oxygen atoms from glutamate 322, threonine 155 and 3 oxygen atoms from water molecules. Carboxylates can act as terminal eta 1, as chelating eta 2 or as bridging mu eta 1 eta 2 ligands. All these 3 types of carboxylate to metal coordination are found in alkaline phosphatase. Eta 1 is shown by aspartate 369, eta 2 is exhibited by aspartate 327 and the bridging mode is shown by aspartate 51. 2 mechanisms have been proposed for the catalytic action of the enzyme alkaline phosphatase. In 1991, Kim and Wyckoff proposed a 2-metal ion assisted catalytic mechanism of phosphate monoester hydrolysis. The proposed reaction mechanism involves 2 zinc metal ions, 2 amino acid residues of the enzymes, arginine 166 and serine 102, and 1 out of the 3 water molecules present in the active site. They arrived at the mechanism based on the structure of alkaline phosphatase with bound phosphate. Alkaline phosphatase functions through the formation of a phosphoseryl intermediate to produce free inorganic phosphate or by transferring the phosphoryl group to other alcohols as shown in the figure. The free enzyme interacts with the dianion of the phosphomonoester ROP to form the enzyme substrate complex EROP as shown here. In this complex, the ester oxygen is coordinated to zinc 1, a second phosphate oxygen is coordinated to zinc 2, and the remaining phosphate oxygens interact with the guanidinium group of arginine 66 by hydrogen bonds. Then serine 102 activated by zinc 2 acts as a nucleophile and attacks the phosphate group of the substrate, forming the covalent phosphoenzyme intermediate EPI and liberating alkoxide. Now a water molecule coordinates to zinc 1 in the position vacated by alkoxide. In alkaline pH, this water gives zinc OH. This activated water hydrolyzes the covalent intermediate EPI into non-covalent enzyme phosphate complex EPI. The dissociation of phosphate from this complex gives the free enzyme. This is the slowest step and therefore the rate determining one. The magnesium 2 ion does not have a direct role in catalysis, but it is important for the full enzyme activity. In 2000, a revised catalytic mechanism called the 3-metallion assisted catalytic mechanism of hydrolysis of phosphate monoester by alkaline phosphatase was proposed by Steck et al. It could explain two uncleared aspects of the 2-metallion catalysis mechanism. These include the mechanism of deprotonation of the hydroxyl group of serine 102 and the role of magnesium ion in the third metal site. 3 water molecules are present in the phosphate binding site in the free enzyme E. Magnesium has a coordination number of 6 and is coordinated to oxygen atoms of aspartate 51, threonine 155 and glutamate 322, and 3 oxygen atoms from the 3 water molecules different from the earlier ones. One of the coordinated water molecules is at a distance of 4.7 angstrom from zinc 2 and 3.1 angstrom from serine 102. This water is coordinated as a hydroxide ion to magnesium and functions as a general base and accepts the proton from O gamma of serine 102. In the EROP complex, the ester oxygen atom is coordinated to zinc 1. 2 oxygen atoms of the ester substrate are hydrogen bonded to guanidinium group of arginine 166. And the 4th oxygen atom is coordinated to zinc 2. Serine 102 occupies the position opposite to the leaving group. The hydroxide ion coordinated to magnesium deprotonates serine 102 by acting as the general base. Then serine 102 performs a nucleophilic attack on the phosphorus atom, resulting in the formation of the covalent enzyme phosphate intermediate EP. This results in the inversion at the phosphorus center and loss of the leaving group RO-. This is followed by the attack on the phosphorus in EP by the nucleophilic hydroxide ion coordinated to zinc 1, resulting in the formation of the non-covalent enzyme phosphate complex EPI. A second inversion of configuration occurs at the phosphorus center. The water molecule coordinated to magnesium now acts as a general acid, donating a proton to serine 102.
Second most important multi-metal zinc based enzyme is liver alcohol dehydrogenase, which is abbreviated as LADH. After studying the alkaline phosphate enzymes, let's now study about the liver alcohol dehydrogenase enzymes, which is abbreviated as LADH. Alcohol dehydrogenase belong to oxoreductase family of enzymes. The transfer of hydride is a redox reaction, but it can also be visualized as nucleophilic attack of hydride on the substrate. If the process is carried out enzymatically, the enzyme provides the binding sites for the two moieties between which transfer of hydride is occurring. Liver alcohol dehydrogenases are one such metalloenzymes. A great deal of structural and mechanistic studies have been performed on the horse liver alcohol dehydrogenases. Liver alcohol dehydrogenases catalyze the reversible dehydrogenation, oxidation of the primary and secondary alcohols to aldehydes and ketones respectively. The overall reaction is shown in the equation.
Liver alcohol dehydrogenase consists of two identical polypeptide subunits, each of mass 40 kilodalton. Each subunit contains 2 zinc 2 ions and binds 1 mole of NAD+. Each subunit of the dimeric enzyme is divided into a coenzyme binding domain and a catalytic domain that are separated by a cleft containing a deep pocket. Both zinc ion are present in the catalytic domain. One is catalytic zinc and the second zinc has a structural role. The catalytic zinc 2 is located at the bottom of a hydrophobic pocket in a deep cleft about 2 nanometer from the surface of the protein. It has a coordination number of 4 and is ligated to 2 cystinate sulfur atoms from cysteine 46 and cysteine 174, one N-epsilon 2 nitrogen of histidine 67 and a water molecule. The coordination geometry is distorted tetrahedral. The structural zinc is close to the protein surface but inaccessible to the solvent. It is coordinated to 4 cysteine thiolates ligands, cysteine 97, cysteine 100, cysteine 103 and cysteine 111 in a regular tetrahedral arrangement. Let us now understand the catalytic mechanism of liver alcohol dehydrogenase. Each monomer of liver alcohol dehydrogenase has two protein domains, the catalytic domain and the coenzyme binding domain, which can rotate freely with respect to each other. Solvent is accessible to the catalytic zinc and the fourth ligand is water in the open conformation. When the enzyme NADH binds, it triggers a major change in the conformation of the enzyme. The binding of NADH to the enzyme induces rotation of one domain resulting in the closed form. This leads to the proper orientation of the groups. The catalytic zinc ion can now bind the substrate, aldehyde or ketone in such a manner that the CH2 group of nicotinamide ring of NADH faces the carbonyl carbon. Thus, catalytic zinc 2 has two roles, geometric and electronic. It gives a specific spatial arrangement to the substrate and polarizes the carbonyl bond. The catalytic mechanism for liver alcohol dehydrogenase is shown here. The coenzyme NAD+ binds to the coenzyme binding domain. At this stage, the enzyme is in its open form. The substrate alcohol enters the cleft between the catalytic domain and the coenzyme binding domain and displaces the water molecule coordinated to the catalytic zinc. The rotation of the domain changes the liver alcohol dehydrogenase enzyme into the closed form. All the water molecules are extruded from the cavity. Then the oxygen bound proton is removed from the zinc coordinated alcohol. This is followed by transfer of hydride from the alcohol CH to the nicotinamide ring. The intermediate involved is a ternary complex of NADH, aldehyde, catalytic zinc. The product aldehyde leaves and the catalytic zinc 2 retains its original coordination by attaching a water molecule. NADH is released and this brings back liver alcohol dehydrogenase to its open form.
Let us now summarize what we have learned in this module. We have discussed the two zinc enzymes, carboxypeptidase A and carbonic anhydride 2, and have noticed that they have only one element, that is zinc, in them. And two more zinc enzymes are there. One is alkaline phosphate and other is liver alcohol dehydrogenase. Both are multi-metal zinc enzymes. We have also studied about their structure, active sites and reactions catalyzed by them. Each active site of the homodimeric alkaline phosphate contains 3 metal binding sites. 2 are occupied by the zinc ions and 1 by the magnesium ion. All the 3 metal ions are involved in the catalytic mechanism. Alkaline phosphate catalyzes hydrolysis of a variety of the phosphate monoesters and also transferylation reactions. Liver alcoholic dehydrogenase, a homodimeric enzyme, has 2 zinc ions in the active site. One is a catalytic zinc and the second zinc has a structural role. LADH catalyzes the reversible dehydrogenation / oxidation of the primary and secondary alcohol to aldehydes and ketones respectively. So after studying this module, you will be familiar with all types of zinc metalloenzymes which have one or more metal besides zinc. One metal plays the catalytic role and the other metal enhance the catalytic activity of the site.